at Bartlett Tree Research Laboratories. Today he will discuss the biology, identification, and management of several common Ganoderma species in the southeastern United States. This webinar is designed to help woodland owners, foresters, natural resource professionals, researchers, and concerned citizens to learn more about shiny-capped Ganoderma species in the United States. And with that, I'm going to turn it over to Dr. Lloyd. All right, Dr. Lloyd, you should be all set.
All right. Thank you very much, Molly. I will
Thank you.
Try to share my screen and get things rolling here. Let me see. How's that?
Yes, we see your title page. You are good to go.
Perfect. All right. Well, thank you for attending this webinar, and thanks for hosting it, Molly and Dave. And glad so many people are excited to learn about shiny-capped Ganoderma species. So just a little more background on myself. I work for the Bartlett Tree Experts Company based out of the Bartlett Tree Research Laboratory in Charlotte, North Carolina. And I focus on diseases of trees and management of them and understanding the biology to better manage them across the United States. The company has offices coast to coast. We mostly deal with residential tree maintenance and healthcare. And that's kind of what my role is as the researcher and technical support on plant diseases. So without further ado, I'll get going with Ganoderma. Just to kind of plug a few things that are coming out, there's a magazine called Fungi Magazine. It's a pay-for magazine subscription, but the edition of Spring 2019, Volume 12, that's coming. It should come out next week or the week after for purchase. We have an article in there that goes over a lot of this. It goes over morphology, and it has a identification key for the Ganoderma species in the U.S. So that should be coming out soon. And then another one that might be more accessible and is available to the public is an extension bulletin put out by the University of Florida Extension Service, an EDIS publication that can be found online, but this is the title, the Lacate Ganoderma of the Southeastern U.S., and it also has a key and some nice pictures and different morphology things that are helpful for identification. So just wanted to plug those. They will be available to you. And before I get going, this is the work that I did on Ganoderma was part of my PhD project at the University of Florida, and lots of people helped me. You can see through this list, but certainly all the people that helped collect these fungi for me so we could identify them and understand them a little better throughout the country. Have to pay a lot of thanks to them, and the funding sources, obviously Bartlett as well as ISA Florida chapter. All right, so let's get into Ganoderma. So I'll be using the term lacate probably off and on. This term essentially just means shiny, and actually it's somewhat redundant because the word Ganoderma, the genus itself, is Greek for meaning shiny skin. And the reason for this is the fungi, which probably most people have seen if you've been working with forestry or just have an interest in trees in general. It's a very common globally distributed genus, and a lot of the species have a shiny surface to their fruiting bodies, as seen in this picture here. There are another group of Ganoderma species that do not have shiny caps. These are the Ganoderma applanatum or aspersum species complex. I'm not going to really focus on those. A lot of these species, the dull-capped ones, are perennial conchs, so they fruit and then continue to fruit from that same structure from year to year. I'm going to be focusing on the annual fruiting bodies species complex that have the shiny cap. So this group of fungi in general, they causes a white rot type of decay, breaking down both the cellulose and lignin of wood, which ultimately weakens the structural integrity. And in a forest setting, that's all well and good. It's recycling nutrients, Decay fungi are great for that, and there's not many things that can decay wood, but fungi are some of these organisms, and they have enzymes to degrade these very complex wood sugars. Ganoderma happens to be able to decay both of them. We call it a primary decay fungus. What we mean by that is that it's one of the few genera of fungi that come in first to do decay. So if you can imagine a forest and you clear cut it, there's a handful of fungi that can come in and start the decay process of those woody tissues and those stumps, and Ganoderma is one of those genera. There's certainly others that do this, but because of that, we often find them in urban settings on living trees and causing pockets of decay or decay of the roots and the large buttress roots of living trees as well. And then that can be somewhat concerning from a hazardous perspective or tree risk standpoint. And through the literature, what we noticed was there was somewhat of a debate or controversy as far as what this thing was doing. Is it considered a disease agent? Is it a pathogen? And what we wanted to do with this research is kind of answer that question: Is it a pathogen, or does it cause disease, or is it just a saprophytic organism that takes advantage of a situation? All right. So there's also other significance culturally with this group of fungi, this genus Ganoderma. It's actually commonly referred to as reishi or lingzhi, depending on where you are in Asia, but it's been used for thousands of years as East traditional medicine. People will collect the fruiting bodies, they will dry them and make teas or tinctures with them, and supposedly it's considered somewhat of a cure-all. In the earliest writings of traditional medicine in Asia, they consider this fungus the mushroom of immortality, and it's considered one of the most superior medicinal herbs, just as a side note. And there's a lot of publications related to this if you want to follow up on that. But no matter if you're talking about the decay fungus or talking about the fungus in terms of its medicinal benefits, there's a lot of problems with the taxonomy with this group of fungi. And in the United States itself, there were several people that have worked on this group of fungi over the years. And this graph is showing 4 people that have worked on this group in North America, starting with William Merle, who in the early 1900s described 16 unique species in North America of Ganoderma that were the shiny cap variety. And over the years, you can see up until 1986 with Robert Gilbertson working on it, the number of species was cut way down to 5 of shiny Ganoderma species in the U.S. And the reason for this is that most people, aside from Merle, thought that Ganoderma lucidum was a species that existed in the U.S. And if you read about this group of fungi, you'll see that Ganoderma lucidum is widespread in the United States, and Merle in the early 1900s said this is not the case. But a lot of species we think Merle identified as unique were lumped into this species Ganoderma lucidum we had thought, and that's what the work we wanted to answer that question of what species actually exist. Is Ganoderma lucidum actually in North America? So this is the probably most up-to-date treatise or book that works with identification of polypore fungi. These are decay fungi that have fruiting structures that have pores on the underside of the cap where the spores are discharged. The North American Polypores, Volume 1 and 2, written in the late 80s by Gilbertson and Rivarden. And you can see this is a picture, and in the top left of this black and white image, you can see a map of the United States. And this is the geographic distribution of Ganoderma lucidum. So you can see it is in the West Coast as well as widespread throughout the east of the Mississippi, and it's quite prevalent. The authors of this book do acknowledge that the taxonomy of the group cannot be stabilized, more work needs to be done, And it was we took the challenge to do the extra work to figure out what species actually exist. So getting into maybe geeking out a little too much here in the genetics realm of things, this was in the early 2000s when DNA technology started to become more commonly used in mycology world. This is one of the first phylogenetic trees or genealogies of Ganoderma, the shiny species around the world. And all I want to point out here is that the red stars on this tree indicate that I guess people are unable to hear the audio. Sorry about that. But the red stars indicate groups or genetic groups that contain the species Ganoderma lucidum. So you can see it's here in Canada. Ganoderma lucidum is also in Europe, in the UK and France, as well as in Korea and South America. So with that in mind, in the US, with that in mind, this is like if I showed you a tree, a genetic makeup of oak trees, let's say red oak trees. You might have, southern red oak in this clade, southern red oak here, here, and here, and it's not the case. So we really need to figure out what these other species are by using good morphology as well as the genetics. So having said that, we understand there's this problem with the taxonomy of what species exist. It seems like a lot of species have been lumped into this one, Ganoderma lucidum. And there's actually a lot of work that has been done using this name in the United States, from looking at it from a taxonomic standpoint, as well as observations of it as a parasite of shade trees. This study was done in New York and looking at it on maples, as well as looking at it from a standpoint of how aggressive it is, a decay organism relative to another, and then the infection pathogenicity of it on grapevines out west. So they're using this name Ganoderma lucidum, but we're really not sure what species they're actually talking about until we delve a little deeper and try to reevaluate some of these isolates. So essentially this. We have failure to communicate, and so we have to understand what species do exist in order to understand their biology and what they mean when we see them in nature. So my work was broken up into two main sections. The taxonomy section, which kind of just cleverly stealing from Eminem, will the real Ganoderma lucidum please stand up? Where I focused on answering the questions, what species of shiny Ganoderma exist in the United States? This was published in an open access journal that you can find online with that title. And then I did some work on other aspects of Ganoderma taxonomy. And in addition, the second part of the research was looking at function, with the general question, are all of these Ganoderma species that exist here, are they all created equal? So I looked at a few aspects of this as well that we'll go over just a few chapters of that today. So just getting right into it, looking at what species of shiny-capped Ganoderma exist in the U.S. So how we went about this was we used somewhat of a citizen scientist approach to make collections, and we had collaborators at different universities as well as mycological societies, and then just looking kind of trolling the online social media with Facebook and this group called Mushroom Observer to make collections. In addition to that, we went to historical records or museums, herbaria that have housed fungi, and we specifically picked two herbaria to study in the southeastern United States. So once we made collections, we characterized the morphology of these individuals. We tried to establish cultures of the fresh material, and then we used genetics to figure out, using barcodes to understand what species, the genetic makeup of them relative to the morphology, to get a better idea of how many species we're dealing with. And this is the current process of identifying and characterizing species of fungi globally to date. So just to go back just straight into the results, we kind of clarified the muddy water, if you will, regarding this group of shiny-capped Ganodermas in the U.S. We identified 13 unique Ganoderma species that have a shiny cap in the U.S. Based on the survey results, you can see in this pie chart, some were more common than others. So Ganoderma curtisii, Ganoderma sessile, and Ganoderma zonatum were the top 3 collected species in our survey. But keep in mind that our survey was highly biased to the eastern United States, and even more so to the southeast. And these 3 species are going to be the most common species in the eastern United States. Ganoderma zonatum really being predominant in Florida. However, it is associated with palm trees, so we think it has the geographic distribution of the sable palmetto cabbage palm. But Ganoderma curtisii and sessile are definitely the most commonly encountered species, and we think that these species are probably 2 that were lumped into the broad group of Ganoderma lucidum over time. So Ganoderma lucidum is a species that is endemic to Europe. It was described in France in the late 1800s, and this species is not native to the United States. So we have used the taxonomy over the years from Europe to say that this is a species we have, but in reality we don't. Now, what that means is that they do share a common ancestor and they have a similar function in nature, but they are different species. This is to give you an idea of geographic distribution of those 13 species. You can see some species are pretty widespread in the eastern United States, like Ganoderma curtisii and Ganoderma sessile. But there are others like Ganoderma sugi that are restricted to the northeastern U.S. and to some degree there are some remote populations in the mountains of New Mexico and Arizona. We're still trying to understand that a bit, but this species has a high affinity to hemlock trees. So in the eastern United States, we find it where hemlocks are grown. Similarly, Ganoderma zonatum is found, we have collections from North Carolina, South Carolina, and Florida. It has been reported in coastal Georgia, but essentially where sable palmetto grows, which are coastal areas in the southeastern United States all the way to southeastern Louisiana, we think we can probably find Ganoderma zonatum. And then there are other species like Ganoderma oregonens and polychromum that are restricted to a western distribution. All of these species cause a white rot type of decay on their respective host tissue. This is our new genealogy of the taxonomy, and I promise you we're going to get through this pretty soon, and we'll move on to some more exciting things as it revolves around the function of these species. But it's important to point out that we can't talk about a group of organisms as one when they are separated, and we have good evidence to support that. We have to understand them and put them in their little boxes so we can understand their biology and what they do better. And this is a good start. So we broke it down. The species that are in black font are species that are found in the United States and that are endemic to the United States. So species like Ganoderma curtisii and Ganoderma sessile, the 2 most commonly collected species in our survey, you can see genetically they're quite distinct from each other, one being down here where the other is up here. The other thing that you can note from that is that Ganoderma lucidum, the species from Europe, is down here. So they're quite distinct from Ganoderma lucidum as well as from each other. So we have done this work now, and now we can start to understand and unravel differences in their biology by studying them in the lab and in nature. So as far as those herbaria were concerned, the 2 collections of Ganoderma that we looked at in the United States at University of Florida and North Carolina State University, and we're grateful for having that opportunity. We looked at collections specifically that were labeled as Ganoderma lucidum in these herbaria. And turns out that we had trouble finding individuals that actually were Ganoderma lucidum. We found none. But the most common species that was found in the herbaria was now identified and reannotated as Ganoderma sessile. So you can see 59% of the collections labeled as Ganoderma lucidum, we have reannotated as Ganoderma sessile. So we think that this species being so widespread and so common, that this species was one that was probably lumped into Ganoderma lucidum for a long time, even though it's quite different. So overall, we found 13 different Ganoderma species in the U.S. That are native here. There were some differences in host groups where some had affinities to hardwoods, others had affinities to pines, and then there was one that had an affinity to palms. And a lot of this information can be found in those documents I referenced in the beginning of the talk. Ganoderma lucidum was not native to the U.S. We did find some collections of this fungus in the U.S. We think that there was probably multiple introductions of this fungus, either on tree rootstock from Europe or through the medicinal fungi trade. And the ramifications of introducing an exotic decay fungus, we don't really know. But it's something that we should think about, and it's not something that we're going to necessarily jump on board and regulate. It's not an emerald ash borer from a tree health standpoint, but we don't know the ramifications of it. So we made that aware. And we had some species that were more common than others, Ganoderma curtisii and sessile being the most common on the East Coast. Okay, so the next thing we wanted to do was take common species that we found in our collections and ask the question, what are the differences in the function? Are all Ganoderma species created equal? And by that, do they have differences in the way that they decay wood, or the rate at which they decay wood? So we took 4 species and looked at them. But before I get into that, a little background information going into it. So broadly speaking, for wood decay, specifically talking about fungi, there are 2 major groups of types of decay. There's a type of decay called brown rot, and there's a type of decay called white rot. So brown rot, it's cleverly named. The wood is left, and it is somewhat of a crumbly or blocky texture, and its color is brown. And this is because lignin is left behind, and lignin has a dark brown pigment. And so these fungi, the brown rot fungi, cannot degrade lignin readily. So when that happens, you get this sort of appearance. Whereas white rot fungi, they degrade both cellulose and lignin. Some of them might decay one preferable over another, but essentially at the end, they will decay everything. And it leaves the wood this white, stringy appearance. So they're breaking down the lignin so you don't get that brown color. Brown rot fungi, typically from a standpoint of tree hazards, can be a little more concerning because this wood, which has been compressed with weight from the crown of the tree and the force, the loads of the canopy, the compression wood is no longer strong, and it gets compressed on itself, and it's very brittle and falls apart. So you can see failures of branches and whole trees from brown rot. Whereas white rot will happen gradually over time. It can also be concerning from a hazardous standpoint, but there's still a little bit of flexibility in that wood, and not all the fibers are gone readily like they are with the cellulose being taken out in the brown rot fungi. So going back to historical studies that looked at Ganoderma lucidum, are studies that use this name to study these fungi and differences in decay, we knew that there were differences. This is on silverleaf oak wood. They inoculated oak wood blocks with different isolates of these 2 species, Ganoderma lucidum, what they called that, as well as Ganoderma sugi, which is the one that occurs on hemlocks. And you can see that in most cases, the isolates that they called Ganoderma lucidum, after a period of, I think it was 120 days, they had percent weight loss of the block about 50%. Whereas with sugi, it was much less. So being able to identify what species of Ganoderma you have could help you understand the aggressiveness of that decay organism. And this is not just true with Ganoderma, this is also true with other decay fungi that we might find in nature or in our urban landscapes. In addition to that, what they also showed was the growth rate. So this graph is showing temperature on the x-axis in Celsius, and they're showing the rate of growth in a petri dish and artificial media. And what you can see is that the temperature, optimum temperature, where the line graphs peak out, for Ganoderma sugi is about 25 degrees Celsius, whereas their isolates of Ganoderma lucidum is really close to 35 Celsius. So getting up into the 80s to 90s temperature range, hotter temperatures, these isolates prefer. So they also were able to suggest that proportionally, compared to decay rates, the faster the organism grew, the more decay it would do. So you could think of a test where you get decay fungi, grow them in the lab, see how fast they grow. It might be able to be a rapid way to screen how aggressive of a decay organism it might be. And that's what they suggested. So their Ganoderma lucidum, as they called them, isolates, grew much faster than the Ganoderma sugi isolates. And in turn, the decay rates reflected that. The Ganoderma lucidum isolates decayed about 50% mass loss, whereas the sugi was much less. So why all this matters is these decay fungi do show up in our urban landscapes, and decay when the structural integrity of wood becomes weaker than the loads that tree has, the forces or the mass that is putting on that, let's say in this case, the root flare of this white oak tree, if that becomes decayed and can't handle the forces or weights of that tree, it's going to fail. And so this is what's concerning. So we have to understand how aggressive certain species are to understand maybe how risky they are when we find them. So what we did was we looked at our collections and we tried to get representative species that were common in the eastern United States, as well as trying to get across different host affinities. So this graph is showing the Ganoderma species that are common on conifers versus hardwoods versus palms. So what we did is we took isolates of Ganoderma, what we called Meredithiae, which occurs on pine trees in the Gulf South. We took isolates of Ganoderma zonatum, which is the Ganoderma species associated with palms in the southeastern U.S. And then two species, Ganoderma curtisii and sessile, that are associated with hardwood decay. We had pure cultures of these fungi. We inoculated these wooden blocks with these respective isolates, and we used wood from pines, loblolly, cabbage palm, water oak, and live oak to look. And we did a randomized complete block design where we grew each of the species onto each of the different types of wood to see if these species, while they do have an affinity in nature for occurring, let's say, zonatum on palm trees, can it also decay a water oak block of wood, let's say. In addition, we wanted to see if there were differences between these species in that ability. Similar to that, we also wanted to look at not only the blocks of wood and the decay that happens, but the chemistry of these different types of wood. We know that some trees are more decay prone than others. Some are more decay resistant. Part of that has to do with active compartmentalization. We know that species like live oak tree in the south is very good at compartmentalizing decay, whereas other trees like southern magnolia are not. But we wanted to look at the chemistry, the amount of tannins and phenolics that are found in wood. They have antimicrobial properties. So what we did is we took wood from these types of trees and we extracted all of the chemicals in the wood, and we grew the fungi on petri dishes with these chemicals and looked at growth rate relative to petri dishes that did not have those wood extracts. This is a block of wood of water oak after 90 days of decay with Ganoderma curtisii. So you can see after 90 days, this wood is quite decayed. It became a sponge. So we know that it works. So this is some electron microscope photos to show you what the different types of wood look like, to give you a baseline. So of controls, these are non-decayed pieces of wood, cross sections. And so this is in the top left corner is the live oak wood. And you can see that the big circles are the xylem vessels. The next size circles that are openings are these larger spongy parenchyma cells, so thinner walled. And then the cells in between are fibers. Pine wood is showing in the next picture here. The larger cells with thinner walls is the early wood, and the thicker cells are late wood. These lines that run through are the ray cells. And this corner is a palm tree cross section. These are vascular bundles. So palms aren't like these other trees. They have vascular tissue throughout the cross section of the trunk, and they have the vascular tissue in these bundles. So the large things are xylem vessels, and then you have phloem tissue underneath that. And then I think these things kind of look like masks with beards. So these larger sections here are the fibrous. They're actually sclerenchyma cells in the beard section, if you will. And then finally, water oak down here, slightly different structure than the live oak, but very similar. So if we look at mass loss, just alone with the different 4 species of Ganoderma relative to the control being the blue bars, and this is after a period of 90 days across all 4 wood types, what we notice right away with water oak is all 4 species can decay water oak pretty consistently and evenly. There was no statistical difference between the 4 species of Ganoderma. But the thing is, we don't find some of these species in nature on water oak, so we don't really understand what that means yet. But we're doing other studies to look at that. If we look at palm trees, not all of the species necessarily decayed the wood the same, but more or less they did. Ganoderma zonatum, if you just look at the number percent mass loss, being about 25 to about 27 or so percent mass loss with Ganoderma zonatum on palm. So it specializes in this, and that kind of made sense to us. Where things kind of got interesting was live oak, and there were differences. Ganoderma sessile, very common species in the South, decayed live oak the most. Ganoderma zonatum did not decay live oak at all. We don't know the reasons behind this, but we think it probably has to do with not only the density of the live oak wood, but also the chemistry behind it. But all this to say, the take-home message for me is that they decay wood very similarly, and it's really a matter of tree species over Ganoderma species as far as how they decay the wood. Some species like water oak we know they're a lot more prone to decay, and they're fast-growing early successional oaks, and the wood tissue is more readily available to decay organisms relative to live oak. So this was something that was good that came out of it. It doesn't seem like there's a whole lot of difference within a species of how they decay, with the exception of live oak and Ganoderma zonatum. But it's really a relative thing about tree host, and knowing the decay profile of a particular tree host is important. To show you what that kind of looked like under the microscope, so you can see this is water oak wood with the 4 different species, and it really degraded everything. It just wiped out all these cells rapidly for each of the 4 Ganoderma species. On palm trees, very similarly, and yeah, we'll skip over that. So going to the effects of the wood extracts on the growth of these fungi, we did notice differences in how these fungi grew with wood extracts. In fact, this one species, Ganoderma meredithiae, which we see on pine trees in the South occasionally, if you look at the control media all the way on the left, this is the culture growth, colony growth of it on when there was no wood extracts present. And then you look at the medias that have the different respective wood extracts in them. You can see it in every instance. Ganoderma meredithiae grew much faster. And we don't know the reason behind this yet, but we think that there are some nutrients that are required for this fungus to really thrive. and probably happened because it has specialized in pines over time, um, and it lost the ability to grow without these wood extracts. But if we look at all four of the species, and this is showing on the y-axis, is the diameter or the colony growth. On the x-axis is the type of media, whether it be the control with no wood extracts being 2% MEA, or one of the wood extract medias. And then each quadrant is the 4 Ganoderma species. So you can see that most species, when they grew on extracts from live oak and loblolly pine, like here in Ganoderma sessile, again in Ganoderma zonatum, and then again for loblolly, specifically on Ganoderma curtisii, it grew less compared to the control. So we think that the resins and some other antimicrobial chemicals in the wood of pine will inhibit decay, whereas others, such as water oak, actually have sugars that will, the fungus will grow the same, with the exception of Ganoderma meridithia. So there are some differences in chemistries as it relates to decay resistance or tolerance in wood. But with all that to say, what we learned from this was that really it's host specific in determining differences in decay of Ganoderma. The Ganoderma species in the lab, at least, seem to decay the different types of wood very similarly. We did not determine that, we could not say, like the authors previously said, that culture growth rate was proportional to decay rate. That was not the case in our study. So, the ideal way of screening these decay fungi, looking at how fast they grow in the lab, can relate to how they decay wood. I don't think it's that simple. So we're going to work on methods of rapidly screening these things to understand their biology. We're going to have to look a little deeper. Wood type was more important than Ganoderma species, like I said, and that's in the nutshell of what it is. We think that the host specificity, so why Ganoderma zonatum in nature is only decaying Ganoderma in palms and not oaks, let's say, that coexist in a bottomland hardwood forest of, let's say, Florida. We think it probably involves more than just the ability to decay. Probably a lot to do with competition. What fungus gets there first, and how can it outcompete the other organisms that will get there next? So moving along to the next question in function, which is, are these things pathogens? So if you scour the internet and try to look at what Ganoderma root rot or Ganoderma decay of trees, how they talk about them in the literature and extension publications like that, there's a lot of confusion out there, not only with the taxonomy of, calling it Ganoderma lucidum, which we know doesn't really exist in most of the United States, but also in what they say about them. So here's one from, not picking on any of these people in particular, this is a problem that, is being resolved currently. This is new. So what Georgia Forestry Commission says, some of these fungi are significantly less harmful than others, but all warrant further investigation. And that's great. That's a very diplomatic answer. But it doesn't really tell us a lot of information about the different species of Ganoderma or other decay fungi. Here's one on Ganoderma lucidum that says, can kill the host tree during a period of 3 to 5 years. But there's no study that actually says that. This is maybe based on observations. We don't really know. And is it implying that it's a pathogen of all of these tree species here in this box? I don't know. Here again from Cooperative Extension article, infected trees decline rapidly and die. The entire crown is affected as leaves fail to emerge. Okay, well, when does that happen? That to me implies that it's a pathogen. And here's one from another group. As soon as possible, remove trees that exhibit conchs on lower trunk and expose root areas. This might not be the case. You might have an area that had a wound, or maybe there's a defect in the root flare, maybe some fused included bark in a certain area that maybe isn't super important for the structure of the tree, and maybe there's just a pocket of decay. But really what we want to see, since we know a little bit about the biology and the taxonomy of these fungi now, is, are they pathogens? Can they kill the tree in 3 to 5 years? So this is this picture in the left, this animated image, is of what palm butt rot looks like associated with Ganoderma zonatum. So we know that it causes, structural damage of the tissue that ultimately leads to the plant dying, and in some cases the tree's failing and injuring property or, God forbid, life. Here is some dieback in this calorie pear. You can see that on the root flares of these trees that actually happen to be buried, you can see Ganoderma fruiting on the root flares. We know these things can decay dead tissues, dead woody tissues as well. So what we kind of have a problem with is understanding, are they killing the tree? Are they taking advantage of a good situation? So going back to those publications, we know that people have talked about this thing as a parasite of shade trees, or implied infections, which implies pathogenicity. And we know that they are considered pathogens of palms in Florida, but also in Asia, where they cultivate oil palms. There's a big problem out there with Ganoderma bonanins on oil palms. And in Florida, there's been some studies that have looked at all kinds of ways. This is from Monica Elliott and Tim Broshot at the University of Florida. They looked at all different ways of inoculating palm trees, young trees, old trees, and using spores versus using colonized wood in different environmental conditions. And it's they had a very hard time, and in fact had really low success to no success of actually inoculating and causing disease. We still call it a pathogen. And don't get me the wrong way, I'm not saying that these things are important and that they're important in tree risk assessments and hazardous situations evaluating them. Decay will lead to structural integrity loss, which will lead to failures. But are they pathogens? Can they infect living tissue? Because the current paradigm is this. You have a tree in the landscape. Joe Weedwhack comes along and damages the root flare. There's a wound created on the tree. Somewhere in the vicinity, there is a Ganoderma fruiting body that's producing spores. The spores are lightweight. They blow in the wind. They land in this wound, and then over time, the fungus starts to decay. And the thought is it can girdle the tissue. The tree declines over time, and then it is prone to wind throw. But the real question here is, are they pathogens? So what we have is a chicken-egg complex. Which came first, the chicken or the egg? So which came first? The stressed tree that led way to decay, and the decay fungi are taking advantage of a situation. Are the decay fungi actually going in and making the tree stressed? Or is it a combination of the two? So to answer this question, what we did is we took the same common species from our collections, Ganoderma meredithiae, zonatum, and curtisii and sessile, and we inoculated trees. We inoculated pine trees, and 1 year we did Pinus tieta, which is loblolly pine, as well as in 1 year we did slash pine. We used jelly palm or Butea odorata as our palm tree to inoculate, and in another study, which I'll talk about, we used Mexican fan palm, and then we used 2 oak species, live oak, and then a red oak group, Shumard oak. We colonized wooden dowels with the fungus for several weeks until it was fully colonized, and then we wounded these young trees. These are trees that are healthy and growing well, and we placed, drilled in, wounded it, placed the infested dowel into the tree, and then let them incubate for a period of 12 months. And we repeated this study twice, monitoring the trees for plant health as well as vigor, looking at wound closures. And so we rated the trees 1 to 3, 1 being healthy, 2 being symptomatic, and what that would look like is maybe some flagging or dieback, and 3 being dead. We looked at wound response. So did the wound close over? And we know in palms this was not going to happen. We did not rate this for palms, but in the conifers and the hardwoods, we rated them on a 1 to 4, where it closed over completely, partially, it didn't, or there was an actual cavity with an external opening present. And long story short is all tree species were rated healthy at the end of the study. There was no symptom whatsoever, and the and the trees, didn't have any issues. In fact, the wounds were either all closed over or partially closed over. We did break down the trees. We cut them out of the root ball, and then we used a bandsaw and split them longitudinally, and we actually were able to measure the area that was discolored. So in this case, this whole area that was discolored, we measured that with a computer program for surface area, and we saw very little differences in discolored area. Shigo's thought of discoloration leads to decay was the thought there. But there really wasn't a whole lot of difference between the controls that were inoculated with just dowels that weren't infested compared to the ones that were inoculated with Ganoderma. So we think the discoloration we saw was just from the wounding itself. When we tried to re-isolate, we failed except for isolates or trees that were inoculated with Ganoderma sessile. This species we were able to recover from the oaks and the pine trees, and although it didn't cause any evident decay, it did appear to be able to be isolated from the discoloration, which does imply that it can infect and potentially lay in a state of dormancy for a period of time, and possibly can lead to decay later on. We don't really know. This is also the only species that produces these survival structures. We call them chlamydospores, and we think probably that is the way that it survived. The tissue inside of the wood, especially living trees and sapwood, is not really an area that these fungi are going to do very well in. It's low in oxygen, it's high in water content, and the plants produce a variety of chemicals that will stop the fungi from actually growing. So, we at the end of this, we don't think these things are pathogens. The tricky situation became with the palms. We know that palms, Ganoderma butt rot of palm is the #1 problem in the green industry of Florida for palms, leading to failures of palm trees and death of them. And virtually all palm species are hosts for Ganoderma zonatum. And we didn't really feel comfortable with our study with the young palms that were maybe still in a grass phase. So we took it a step further, and we got some Mexican fan palms, and we inoculated them, but we used 2 different new methods. So instead of just wounding it and putting 1 dowel, we knew the vascular tissue of the palms would survive if we took out a small section of the palm. So you can see in this picture, we used a saw to cut a wedge out of the woody trunk of these palm trees, or we drilled into the tree with 8 wounds and 8 dowels that were infested with Ganoderma zonatum, evenly spaced around the trunk of these trees. And we tried to inoculate near where the roots were emerging on the trees. For the ones where we took the section out, we standardized by weight infested dowels and replaced that void with dowels, as you see in this picture. And then in both cases, we wrapped the palm trees with burlap, and then we kept them moist in the shade as well as under irrigation periodically during the summer months. Again, over the period of 12 months of monitoring this, we had no palms die, and in fact, we couldn't re-isolate the Ganoderma when we tried to isolate from cross sections. So all this to say, we don't feel comfortable to call these things pathogens of healthy trees, especially not from fresh wounds. The internal discoloration was from a wound response, not from the fungus itself. And the sapwood doesn't seem to be an inhabitable place by these fungi. So we know that Ganoderma species are not good competitors in soil. We probably think they aren't maybe in wood as well. We're not really sure. In pathology, we call these things infection courts, the area that we inoculate. And it's possible that the infection court that we used, i.e. the fresh wound of the tree by drilling or cutting out a section of the palm trunk, the fresh wound might still be inhabitable, not inhabitable by the fungi. It's possible that we need to look at old wounds or wounds that have desiccated tissue, and maybe that's where these fungi get in, and maybe they act more as latent opportunistic saprophytes, which is our current thought. But it doesn't mean that these fungi can't be in the trees. And in fact, before there's any decay present. There's a few studies that have come out that have looked at that, looked at decay fungi present in healthy trees with no decay present. And they've done surveys using DNA technologies to identify fungi that cause decay of wood in healthy trees. And they found an abundant amount of decay fungi that are present and living as endophytes. So I really like the title of this paper, Do All Trees Carry the Seeds of Their Own Destruction? And what they're referring to is these decay fungi that are laying kind of in a dormant state in trees, like we saw with Ganoderma sessile. It infected, but it did not seem to cause any decay. Is it going to be present in that tree? And when the tree goes south for one stress reason, construction damage, a drought year, whatever it may be, and that tree, the wood desiccates, now that fungus is present and can start doing the decay process. So this is the new way of thinking that we're thinking is probably realistic. There's been some papers published on this for hypoxylon, canker on oaks, and that it is an endophyte on trees. And it's probably the case for other decay fungi as well. So the new life cycle is this. We know that Ganoderma can decay dead tissues. We know it can be found on old stumps as well as dead trees. But there's a little bit of gray area on when we're dealing with it in healthy living trees. So a wound probably has to be present, and there probably has to be somewhat of a stress factor over time. So this decline aspect, what's causing that decline, there's probably a myriad of things that can do this. Chronic stresses like root disturbance, soil compaction, drought, etc. But this has to happen for that tissue to become desiccated. Once that tissue is desiccated, the fungus can then thrive and start to decay, and we can find it in pockets. As that pocket forms an external cavity on the tree, its tissue on the perimeter interior can become desiccated. And so it progresses over time, much like a disease, but over a much longer period of time. And this is kind of what we are proposing as how these decay fungi work in nature. We're looking at new ways to test this, looking at different stresses. So one study that we've done is we've stressed trees by topping them, and we've inoculated them. So we're going to try to determine if trees that are stressed are more easy to inoculate and actually cause decay than trees that were not stressed. And we need to look at other things as well. We need to continue to study Ganoderma zonatum on palms and to study that aspect of it, which you can read about in some of the papers we mentioned earlier. And continue to look at other decay fungi. Let's get out of the Ganoderma realm and start doing this for other common primary decay fungi that we can find on living trees to understand what they mean to help us with tree risk assessments. But with that, I've talked enough. I think at this time, I think Molly will probably come on board and ask some questions.
That's right.
There probably are some, and hopefully we can do our best to answer those. But yeah, thanks for listening, and hope you're excited about Ganoderma.
Excellent. Andrew, Dr. Lloyd, thank you so much for that presentation. That was packed with information, and it does kind of look like they have beards. Yeah. So at this point, we're going to open up the presentation for questions from the audience. Because this presentation was so packed, we're only going to have time for a few. And then after that, stay tuned. I'm going to be pushing out the redirect link, which will send you to the quiz that you will need to take to get those continuing education credits. All right. Dr. Lloyd, can you hear me okay?
I can.
Okay, excellent. All right. So first question from somebody was, is APHIS aware of this imported decay fungi from? Yeah.
Well, yes, I did. I have shared the publication and my dissertation with the mycologists at APHIS. They are aware of it being present. Again, I don't think that there's a immediate concern as far as something like emerald ash borer or an Asian longhorn beetle or a laurel wilt situation. I don't think this thing is going to necessarily get out there and do a lot of things, but in the grand scheme of things, it has been made aware, and I think regulating, especially the medicinal fungi industry, needs to happen and result from these studies, because people are selling species from all around the world into this, and they're being introduced in this country. People are growing them at their houses. When they're spent or done fruiting, they're probably throwing them in their compost pile, and there's a chance that they can escape. And what that can mean is it can displace some of our native species. Potentially, these could be more aggressive decayers. They can also potentially outcompete some of our other native decay fungi. And so from a biodiversity standpoint, it could be somewhat concerning. They are aware of it. Like I said, I don't know. It's probably not a priority, especially with funding continually being cut and that sort of thing. But yes, they are aware.
Okay. Quick question here. What species did you say is found on eastern hemlock?
On hemlock, that would be Ganoderma sugae.
Sugae. Okay.
Psugae.
All right. Where were the pathogenicity tests done? As in what part of the country, and did you see a difference in the rates seasonally?
Oh, good question. Yeah. So they were done in Gainesville, Florida. And we, one repetition of the experiment we inoculated in the springtime. And then the other repetition of the experiment we inoculated in the fall. We didn't really see differences between the two other than wound response. It seemed like the ones that were done in the growing season, there was a much quicker wound response than the one in the fall. But as far as decay went and re-isolating the fungi, there really wasn't a difference there.
Okay. Is a tree with Ganoderma more susceptible to other insects and diseases?
Great question. Yeah, so if decay is present in the tree, it's possible, depending on the extent of that decay. If there's an excessive amount of decay in the tree will probably be in a state of decline. And there's a whole slew of secondary organisms, bark beetles, ambrosia beetles, that are going to be attracted to that stressed or declining tree. So certainly it might not be causative, but might be correlated. And it's really correlated to the decline of the tree, that these things all exist together.
Okay. And sort of as a follow-up, like what can be done? Do antifungal treatments like trunk injections work to prevent or cure Ganoderma?
Great question. No. So right now we have no chemical treatments that can be applied legally according to labels, as well as effectively to stop decay, unfortunately. There are a few biologicals that have been used. It seems like the results of these studies, these are trichoderma species, products like that that contain those species. There's variable results in the research, and we haven't found consistent things with it. The best thing that we can do when we find decay is consult with a certified arborist that has a tree risk assessment qualification from the ISA. So the ISA has a qualification course. One prerequisite for it is having a certified arborist license through ISA. But then you can sign up for the course. It's a 2.5-day course. They teach you all the ins and outs of the current industry standard for tree risk assessments. And so what that involves is, from the standpoint of decay, which decay being one type of defect that might exist, you would determine the extent of that decay and relative to the crown or the loads of the canopy, and determine a mitigation option. Occasionally, if the decay is extensive, and there are what we call targets, or pieces of property or people walking by on the regular, if they're at risk of being injured by a part of the tree or the tree itself, because there's a lot of decay, one mitigation might be removal of the tree. But if there's only a small portion of the tree that has pockets of decay, there's other mitigation options, like reducing the load of the tree. So this would be like pruning, to reduce the amount of weight that's actually being pushed and compressed on that area that has decay. And then what we would like to focus on is really looking at improving the vitality of the tree. And this can involve decompacting the soil with aerification using an air spade, which is a tool that uses compressed air to really decompact soil, and the addition of organic matter as well as fertilizing according to a prescription or a soil analysis to really boost the health of that tree. If we can boost the root system of that tree, theoretically, if the tree is a good compartmentalizer, it'll have more reserves and energy to compartmentalize. So that's the other thing, is knowing your tree species. But when in doubt and you're a homeowner or you're not qualified to do tree risk, it'd be a good idea to reach out and find a qualified person to do that, to really come up with a game plan. Because I think at this point it's going to be a case-by-case basis on what the mitigation options are for your situation.
Okay, that's an excellent answer, and actually answered the questions of about 3 or more others from folks. So guys, I think that about does it. We've run out of time. If you scroll up, I shared the redirect link earlier. I'll do that one more time now. If you want CEU credits, it's very important that you follow that. And I'm sure, and Dr. Lloyd, if you could hear, everybody is giving you a round of applause today. Thank you so much for joining us. That was an excellent, excellent presentation.
Great. Thank you very much for having me.
All right. Have a good one.
Yep, you too.
Bye-bye.
Bye-bye.